Hello guys, welcome to this YouTube channel. A good day to everyone watching this video. So in this video, I am mainly going to discuss about the GATE Excel Biochemistry previous year's questions of the year 2022. So let's get started. So the first question, which of the immune cells listed below are agranular? So the options are eosinophils, mast cells, monocytes and T cells. So clearly we know that the polymorphonuclear leukocytes, they are granular, so which include the eosinophils and mast cells. However, they have asked which are agranular. So for that the answers are monocytes and T cells. So the answer is going to be option C, which are RNS, which is monocytes and T cells. So that's a pretty simple question. So moving on to the next question. Okay, which one of the following enzymes is located in the outer mitochondrial membrane? So if you look at the options, you have three main enzymes of the TCA cycle, which are citrate synthase, fumarase, and succinate dehydrogenase. So these enzymes are located in the mitochondria. However, monoamine oxidase is an enzyme that is actually located in the outer mitochondrial membrane. So the correct answer here is monoamine oxidase. So in this type of questions, it is better to go by, deductive reasoning, wherein you know that the other three enzymes are the enzymes of the TCA cycle, and hence the one which is left out is going to be the enzyme that is located in the outer mitochondrial membrane. So that's a pretty smart way to answer this question. Moving on to the third question. Which one of the following statements about the DNA polymerase 3 of E. coli is not correct? So the options are: it catalyzes Nick translation, its absence is lethal to E. coli, it synthesizes a complementary DNA strand using a single-stranded template, and it possesses 3' to 5' exonuclease activity. And here the correct answer is it catalyzes Nick translation. DNA polymerase 3 of E. coli does not do this job. However, its absence is lethal to E. coli, and it synthesizes a cDNA strand using a single strand template, and also has a 3' to 5' exonuclease activity for proofreading. So the correct answer here is option A. So these are few facts that are vital to remember so that it becomes easier to answer these questions. Moving on. Which one of the following compounds is not a translation inhibitor? So all these are antibiotics, and it is again very important to know the mechanism of action for various antibiotics. So I keep telling this in many of my previous videos as well, that antibiotics are a pretty frequently tested question in various biology competitive exams. So here the correct answer is rifampicin, because it mainly inhibits the DNA dependent RNA polymerase. It inhibits DNA dependent RNA polymerase, especially by binding to the beta subunit of the RNA polymerase. So the correct answer here is rifampicin, because it doesn't inhibit translation but rather the transcription process. So the correct answer here is rifampicin. Moving on to the next question. A dye was allowed to undergo migration on a chromatographic paper using a solvent. The dye and the solvent front migrated 5 cm and 20 cm respectively from the point of origin. So what is the retention factor that is rounded off to 2 places of decimal for the given dye? So it's a very, very simple question. So retention factor is given by the formula: the distance moved by the dye, distance moved or traveled by the dye, divided by the distance moved by the solvent front. Solvent front. So substituting these values, you will get 5 by 20, which is 1 by 4, which is 0.25. Very simple question. So the retention factor here is 0.25. If you just know this formula, then it's a very easy question. Moving on to the next question. Wait, I think it's the fifth question. Yeah, this one. Yes. The pKa values of the carboxylic and amino groups of an amino acid with a non-ionizable side chain are 2.17 and 9.13 respectively. So the isoelectric point of the given amino acid is. So it has to be rounded off to 2 decimal places. So we know that by formula, the isoelectric point is given by the formula pKa1 plus pKa2 divided by 2, provided the given amino acid has a non-ionizable side chain. If the amino acid has an ionizable side chain, then you have another pKa value, and accordingly, based on whether it's an acidic or a basic amino acid, you have to incorporate which two pKa values that you need to choose. So here it's pretty simple that it's a non-ionizable side chain. So the pKa's correspond to the pKa of the carboxylic acid group and the amino group. So here, substituting the values, you will get 2.17 plus 9.13 divided by 2, and the answer comes out to be 5.65. So the isoelectric point of the given amino acid is 5.65. Again, it's a very easy question to score full marks. Moving on to the next question. The number of ATP molecules required for the complete assimilation of one molecule of CO2 in Calvin cycle is. So basically, if you have good knowledge in plant biochemistry, then this particular question is pretty easy. If you can remember the overall reaction of the Calvin cycle, which is 3CO2 plus 6NADPH plus 9 ATP gives G3P, which is glyceraldehyde 3-phosphate, plus 6NADP plus 9 ADP. So here, for 3 carbon dioxide molecules, you require 9 ATP. So for 1 CO2, you will require 3 ATP. So here, the number of ATP molecules required for the complete assimilation of one molecule of CO2 in Calvin cycle is 3. So again, it's a very simple question if you know the formula, if you know the overall reaction of the Calvin cycle. So people who are good at botany and who have some fair knowledge in plant biochemistry, it's a pretty easy question to solve for them. Moving on to the next question. The absorbance of 5 into 10 power minus 4 molar solution of tyrosine at 280 nanometer wavelength is 0.75. The path length of the cuvette is 1 centimeter. So what is the molar absorption coefficient at the given wavelength in molar inverse, centimeter inverse, correct to the nearest integer? So again, by Beer-Lambert's law, we know that A is equal to epsilon Cl. So A is absorbance, epsilon is the molar absorption coefficient, C is the concentration, and L is the path length of the cuvette. So substituting these values, you get A as 0.75 equal to epsilon, which we need to find, which is the molar absorption coefficient, and concentration is 5 into 10 power minus 4 molar into L, which is 1, because they ask the epsilon in terms of centimeter inverse. So you can just keep it as 1. So epsilon here will be 0.75 divided by 5 into 10 power minus 4. Oops. Yes, that is equal to 0.75 into 10 power 4 divided by 5, which is 7500 divided by 5, that is going to be 1500 molar inverse centimeter inverse. So it's a very direct question of the Beer-Lambert's law, and the answer here is 1500 molar inverse centimeter inverse. Moving on to the next question. Yes. So the filamentous photosynthetic algae were placed on a microscopic slide and illuminated with light of different colors as illustrated. So here you can see blue, green, yellow, and red. So the bacteria that are known to migrate towards the region of high O2, high oxygen, were also added uniformly on the slide. So which one of the following options illustrates the distribution of bacteria along the length of the microscopic slide after illumination? And here if you look at the options, so here the graph represents the y-axis of the graph represents the relative bacterial concentration, and the x-axis represents the region corresponding to the color of illumination. So here the correct answer is A. That is because this is a microalgae, so it's a phytoplankton. Basically, it's usually in underwater, and due to the pigment composition and resulting absorption spectra, the red and the blue light here actually produces higher growth rates and biomass accumulation than the yellow and the green light for the microalgae. So this is basically a fact. Usually most botanists would know this. So that is why the bacteria which are known to move to the region of high oxygen levels, they tend to go towards the blue and the red region, and they are their concentration is pretty less in the green and the yellow region. So basically the optical properties of the microalgae determine the underwater light distribution that directly correlates with the growth. So that's the reason why this is the correct option. So if you have done some good courses in botany, then this is something that you would be aware of. Exactly. So this was asked in the 2022 paper. Moving on to the next question. Yes. So two RNAs shown below were used separately as templates in an in vitro translation system which can generate proteins in all possible reading frames. So RNA1 has 5' AG repeated n times to 3', and RNA2 has 5' AAG repeating n times to 3'. So the RNA1 translated product contained arginine and glutamate, and the RNA2 translated product contained arginine, glutamate, and lysine. So which one of the following codons directs the incorporation of arginine? So first of all, let's expand this code basically. So write it as AG repeating n times till 3'. And then here RNA2, this is basically RNA1. So for RNA2, it is 5' AAG repeating n times up till 3'. Okay, so now it can be read in all possible reading frames. So what are the possible reading frames for the first RNA? So that will be AGA as one reading frame, or you can have GAG. So these two are the only possible reading frames. For the second one, it can be either AAG or AGA or GAA, right? So AAG, AGA or GAA, this three. Okay. So now if you look at this, so the first RNA codes for arginine and glutamate, and the second RNA codes for arginine, glutamate, and lysine. So if you compare these two reading frames, so which codon is common in both? That is AGA is common in both, and as a result, the answer is B, which is AGA, that is coding for arginine. So this codon directs the incorporation of arginine because that is common in both, and both of these RNAs translation product contain arginine. Okay, so that's the reason why it is the correct answer here is AGA. I hope it is pretty clear. Yes, so it's just basic logic of how codons are read and how the common sense that we apply in figuring out the common codon between the two RNA frames. Anyways, reading frames. Moving on to the next question. Which of the following statements about endogenous synthesis of insulin are correct? So the statements are: insulin is synthesized as a pre-proinsulin. The pre-proinsulin is converted to proinsulin. The single-site cleavage of proinsulin eliminates C chain, and mature insulin consists of disulfide linked A and B chain. So the correct answer here is option B, that is P, Q, and yes are correct. Of course, the single-site cleavage of proinsulin eliminates C chain. That's not correct. However, the other three options are correct. So it is important to know how insulin is produced endogenously. And, insulin is a very famous protein that is usually tested in most of these biology exams, and since I'm only discussing the previous year's questions, I'm not going too much into details, but it's important that you go through how insulin is produced, especially with the A, B, and C chains, and especially how the recombinant DNA technology also utilizes this to produce a mature insulin peptide. Okay, moving on to the next question. Which one of the following enzymes converts testosterone to estradiol? It's a very direct question, and the option is answer A. Option A, which is aromatase. So it's a very basic question that people reading about steroidal hormones know. So the correct answer here is aromatase. That is the important enzyme that converts testosterone to estradiol. Moving on to the next question. Purification of 6X histag protein using NINTA column is an example of very direct question, that is affinity chromatography. So proteins that have a 6X histag are retained by affinity chromatography, which has this NINTA column, and then a different elution buffer has to be added to, elute these, the protein of interest that has the 6X histag. So it all comes with the recombinant DNA technology to produce the protein of interest with 6X histag. Moving on to the next question. Which one of the following carbohydrates has or have a beta 1,4 glycosidic linkage? So this is a multi-select question. So we need to be pretty careful about the options that we choose. And the correct options are A, B, and C. Maltose has an alpha 1,4 linkage. So all these three polysaccharides and disaccharide, of course lactose, they have a beta 1,4 glycosidic linkage. Moving on to the next question. Which of the following statements about IgA is or are correct? So, it is secreted into colostrum, it is transported across the cell by transcytosis, and its secretion is facilitated by poly-Ig receptor, and it primarily exists as a dimer in the serum. So, the correct answers are options A, B, and C. Of course, it is secreted into colostrum in the mother's milk. It is transported across the cell by transcytosis. Yes, if you can remember the immunology diagrams where IgA is clearly, transported across the gut epithelium via transcytosis, and its secretion is facilitated by poly-Ig receptor. Yes, that is true. And however, in serum, it is mainly in the monomeric form. Only in secretions like sweat, saliva, mucus, etc., it exists as a dimer. So this is incorrect. So, however, the other three statements are correct. So those have to be ticked. Moving on to the next question. The standard free energy changes for conversion of phosphoenol pyruvate, that is PEP, to pyruvate and ATP synthesis are shown below. So you have this reaction. However, remember these are standard delta G naught. So you should remember these are not delta G but the standard free energy change. The starting concentrations of PEP, ADP, pyruvate and ATP are 25, 25, 50 and 50 millimolar respectively. So the value of the universal gas constant R is 8.315 joule mole inverse Kelvin inverse. The actual free energy change in kilojoule per mole for the reaction, that is PEP plus ADP gives pyruvate plus ATP, carried out at 37 degrees Celsius will be what? This is a basic thermodynamics question that we should all be knowing how to solve. So basically, if you add up these reactions, if you add up these two reactions, you get this. So in that case, when you add up two reactions, the standard free energy change will also get added up. So that will become minus 61.9 plus 30.5. So that will be minus 31.4. However, by formula, we also know that delta G equal to delta G naught plus RT ln the concentration of the products, basically it's the reaction coefficient, divided by the concentration of the reactants, right? So if you substitute these values, that is minus 31.4 plus 8.315 into T. So T here is given as 37 degrees Celsius and we have to change it to Kelvin. So how do you change it to Kelvin? You add 273 to it. So that will give you 310 into ln. The products are pyruvate and ATP and they have a concentration of 50 and 50 millimolar respectively. So you multiply 50 into 50 divided by, since everything is in millimolar, I'm not including into 10 power minus 3 because that's going to get cancelled. And if you look at the reactants, which are PEP and ADP, they have a value of 25 and 25. So that comes to divided by 25 into 25. So ultimately, you will get this as minus 31.4 plus 8.315 into 310 into ln 4. So this, however, is in joules. You need to make it as kilojoules. So into 10 power minus 3 kilojoules. So make sure that is mentioned. So that will be minus 31.4 plus 2.579 if you do this entire calculation. So the final answer comes to minus 28.821. That is kilojoule per mole. However, they have asked the answer to be rounded off to, I guess, one decimal place. So in that case, you can mention it as minus 28.8 kilojoule per mole. So that is within the accepted range of what is mentioned in the answer key. Alright, so I guess it's a very simple question to solve if you know this basic thermodynamics formula. Alright, moving on to the next question. The dissociation constant for a receptor-ligand pair is 0.25 into 10 power minus 7 molar. The ligand was added to a solution of the receptor such that the receptor was 50% saturated at equilibrium. Assume that the receptor has one ligand binding site. the concentration of the free ligand at equilibrium in nanomolar, correct to the nearest integer, should be. So first of all, they have given the dissociation constant. I am writing it as Kd, which is equal to 0.25 into 10 power minus 7 molar. So when you talk about dissociation constant, you have to talk about the receptor ligand complex dissociating into ligand plus receptor, right? So this Kd is given by the formula, of course, the products by reactant by Lr, right? So that is 0.25 into 10 power minus 7. Now, if you carefully observe here, you see that the receptor was 50% saturated. When the receptor is 50% saturated, then what happens? The concentration of receptor ligand will be equal to the concentration of the remaining receptor at that given point when it is 50% saturated. So in that case, this and this gets cancelled. So thus, the concentration of the ligand will be equal to 0.25 into 10 power minus 7 molar. However, they have asked the answer in nanomolar. So that will be 25 into 10 power minus 9 molar. So that will be 25 nanomolar. So the full answer, the correct answer here is 25 nanomolar. Okay, this is also very. Moving on to the next question. The half maximal velocity of an enzyme catalyzed reaction was found at a substrate concentration of 0.5 into 10 power minus 6 molar. So this enzyme follows Michaelis-Menten kinetics. So in the presence of a competitive inhibitor, the half maximal velocity was found at a substrate concentration of 1.5 into 10 power minus 6 molar. So given that the enzyme inhibitor pair has a dissociation constant of 2 into 10 power minus 7 molar, the concentration of the competitive inhibitor in micromolar rounded off to 1 decimal place would be what? So first of all, this is Michaelis-Menten's kinetics. So it is very important that you know how to use this Michaelis-Menten kinetics in biochemistry problems. It's going to be a sure shot question in any gate paper or any competitive exam, biology competitive exam. So here, at half maximal velocity, so at V max by 2, we know that substrate concentration is equal to Km if there is no inhibitor. And here, at Vmax by 2, the substrate concentration was found to be 0.5 into 10 power minus 6 molar. So that is equal to Km. This is without inhibitor, right? However, with inhibitor, so this is with inhibitor, let me write it as I. With inhibitor, at Vmax by 2, the substrate concentration was found to be 1.5 into 10 power minus 6 molar, and this is equal to alpha into Km because you have a competitive inhibitor here. So when there is a competitive inhibitor, there is another term called alpha, and this alpha is given by the formula 1 plus concentration of the inhibitor divided by the dissociation constant of the enzyme inhibitor pair. Okay, so first of all, we should find out what alpha is. So we know that alpha into Km is 1.5 into 10 power minus 6 molar. That is equal to alpha into Km is 0.5 into 10 power minus 6 molar. So now alpha is equal to 1.5 into 10 power minus 6 divided by 0.5 into 10 power minus 6. So that gives you 3. Alright. Now substituting in the equation, we have 3 is equal to 1 plus concentration of the inhibitor divided by the dissociation constant of the enzyme inhibitor pair. So that is Ki, which is given by 2 into 10 power minus 7 molar. So now we have I, concentration of the inhibitor, will be equal to 2 into 10 power minus 7 molar into 3 minus 1. If you subtract here, you will have 2. So that will be 4 into 10 power minus 7 molar, which is also equal to 0.4 into 10 power minus 6 molar, which is 0.4 micromolar. So the correct answer here is 0.4. I hope it was a pretty easy question, but you should know this particular alpha formula here. Otherwise, it'll not be easy to solve this question. And also, you should know what Michaelis-Menten's kinetics is. Moving on to the last question. A 40 times diluted sample of single-stranded RNA gave an absorbance at 260 nanometer of 0.01. The concentration of the single-stranded RNA before the dilution in microgram per ml was, correct to the nearest integer. First of all, before I start solving this question, you need to know one particular fact in mind, that is, for a 1 centimeter path length, for a 1 centimeter path length, the absorbance that is of optical density or absorbance at 260 nanometer is equal to 1 for 40 microgram per ml solution of RNA. So this is very important. So for 40 microgram per ml, the absorbance is 1. So for 0.01, what is the concentration? That will be equal to 0.4, right? For 0.01, if you do direct proportion, the concentration would be equal to 0.4 microgram per ml. However, this one is 40 times diluted, right? So what is the original concentration? So original concentration will be 40 times 0.4, that will be 16 microgram per ml. So the correct answer here is 16. So with this, I am completing the discussion of the answer key with all the steps for all the biochemistry questions of 2022 GATE Excel paper. Hope you benefited out of this. Have a nice day and let us see in the next video for 2023. Thank you. Bye.